Hello everyone, I'm Jia Song, come from China. Today I'm going to talk about the agricultural nanotechnology for plant immunity. For nanotechnology, as we know, the search for effective alternative methods of plant disease and control is mandated by the need to reduce or eliminate non-target effects on both humans and the environment. Nanotechnology in agriculture has emerged as a new tool to create and restructure the materials at the molecular level. The molecular nanotechnology involves building organic materials into defined structures, atom by atom or molecule by molecule, often by self-assembly or self-organization. The application of nanotechnology in agriculture has gained an interesting attention in recent years. Biologists are actively engaged in the synthesis and investigation of organic nanomaterials, including different kinds of nanoparticles possessing unusual optical, physical, and biological properties. The possible uses of nanotechnology in agriculture are being explored. Precision farming, for example, along with nano delivery systems, is becoming the new industrial revolution in agriculture. Then there is great potential for nanoscience and nanotechnology in providing state-of-the-art solutions for various challenges faced by agriculture and society today. Nanoparticles can serve as magic bullets containing bioactive substances from antagonistic fungi that can enable effective penetration through cuticles and tissues, allowing the slow and constant release of the active substances. The most popular chips of nanomaterials being used for biocides delivery are nanospheres, nanocapsules, and nanogels. As nanotechnology can provide green and efficient alternatives for the management of disease and insect pests in agriculture without compromising nature. Nanotechnology has great potential in agriculture because its applications can enhance the quality of life, especially in food crop production systems. Nevertheless, although it may play a very important role in the development of any nation, nanotechnology should be carefully evaluated as is done with any new technology. Some nanoparticles have been formulated to contain pesticides in colloidal suspension or as powders at the nano or micro scale. These preparations have advantages such as increasing the stability of active organic compounds, systematic activity, synergism, and specificity, and reducing the foliar salting and leaching. The amount of pesticide dosage, number of applications, human exposure to pesticide, and environmental impact are reduced. So nanoformulation have been employed not only for synthetic insecticide, but also in alternative products such as natural products like herbal extracts and also microorganisms to control insects. Current issues like climate change, urbanization, sustainable use of natural resources, and runoff and accumulation of toxic pesticide, herbicide, and fertilizer need to be addressed immediately. However, a few research have reported the use of nanocarrier systems in agriculture. The use of bioactive compounds from different ketamine species has been proven to be an effective antifungal strategy against several plant pathogens by the research of Soiton 2001. The construction and characterization of copolymer nanoparticles loaded with bioactive compounds rather than toxic pesticides are needed. So for the research of nanoparticles from ketamine species, at the year 2016, Hui Li Ta and Kasim Soitong used to report that the effects of nanoparticles loaded with ketamine globosome KMITL species the extracts against leaf spot of rice variety. And also ketamine cubium CC3003 also can control the leaf spot of rice in Cambodia, which caused by the Coraria lunata. As this figure shows the nanoparticles from ketamine globosome, actually you can see the color is the yellow color in the nanoparticles. And in vitro test, the pathogen in PD media, we can see the control is normal spore, but also in treatment by nanoparticles, we can see a lot abnormal spore as the shape is changed or the size changed. And also the reported showed the spore suspension of ketamine cubium and also nanoparticles of ketamine cubium give the high disease reduction of leaf spot of rice. And you will compare with the chemical fungicide tebuconazole. The nanoparticles carries 50% of disease reduction. And also at the year 2017, Somli Vilawa and Kasim Soitong also reported the application of a new bioformulation of ketamine cubium for biocontrol of Corydodicum gliosporate causing coffee as lactose Arabica variety in Laos. And also the application of nano-lutulino, nano-detoxin, and also tested against the Corydodicum gliosporate for coffee and Tachness disease. The application of this bioformulation of ketamine cubium and nanoparticles to reduce the coffee and Tachness was reported for the first time in Laos. And also you can see the coffee and Tachness symptom on the leaf, and you get the pure control of pathogen and coryndia in the upper figure, and in the down after do the pathogenicity test prove the pathogen can cause disease of coffee. And then the effect of bioformulation of ketamine cubium on control of leaf antagonists caused by Corydodicum gliosporate in coffee variety of Arabica in Laos, as shown in the field test. The nano-decotoxin and nano-lutulino also give the higher disease reduction, as higher as 54% and 46%. So at the recent time, I also researched about the nanoparticles from ketamine species against Megaphoti oryzae causing rice blast, and already published 2 papers. Because for the rice blast, China is the world's top producer and consumer of rice, and also in Asia is the main food of human. But the rice blast is a harmful disease that can affect rice crop. And even in Vietnam, the case is the rice blast disease decreased the yield higher as 93%. And you can see the photo of rice blast disease. And so we also isolate the pathogen as the figure and identify as Megaphoti oryzae. You can see the photo of the coryndia photo is 3 cell and partiform chip. So after the pathogenicity test, the pathogen Megaphoti oryzae gives the disease index higher as level 8, followed by the International Rice Research Institute standard. And so I have already done extract different kind of metabolites from ketamine species. You can see as a photo, the first upper photo shoot is the extracts and also pure compound. And after that, we loaded as nanoparticles as the figure shoot, and they formulate as the liquid formulation. And also this figure showed the the nanoparticles. After we get the nanoparticles, we also doing the scanning electron microscopy to check the size of particles. The size is average is around 400 nanometer. Actually, all the kind of nanoparticles we have already done the checking the size. And also we tested in vitro on the RFA media for culture magnetron proteolysis. And then you can see the photo we can in control plate. We can see the only normal spore, but in the treatment by nanoparticles, we found a lot of abnormal spore. You can see the photo shoot is the cell wall of spore was destroyed, and you can see clearly we test different kind of nanoparticles. This can destroy the spore of magnetron proteolysis. And also the nanoparticles can inhibit the spore production of magnetron proteolysis in the media, and it's inhibit higher as 81% of the spore production. And also the nanoparticles were tested in pot experiment. As the figure showed, we tested in 3 weeks sealing on the rice and then treated inoculated pathogen and treated with nanoparticles, the liquid formulation. And after 5 days, we checked the disease index and collect the data of plant and root. As the figure showed, the treatment 1 is non-inoculated control. Treatment 2 is inoculated control. You can see the disease index as higher as 8 also. But the treatment 3 to treatment 10 is the one we treated with different kind of nanoparticles. The disease index is in average around level 5, but the treatment 11 is chemical. Even compared with chemical, our nanoparticles can give the high disease control also, especially treatment 3 is nanoparticles CG. So, and other data like chlorophyll content and plant fresh weight, dry weight, and root also collected. As we showed in this table, the disease reduction, nanoparticle CG and EA higher as 47-44%, and the chemical gave the 63%. But at the same time, our treatment have the higher fresh weight, dry weight of both plant and root, even compared with the non-inoculated control and chemical control. As the figure also can directly see the plant fresh weight, dry weight is in the treatment by nanoparticles have more high plant fresh weight, dry weight, as also in root also get more high fresh weight and dry weight. And at this after this, we also check our nanoparticles can induce the plant immunity. We check him by the phytoalexins, which produced by plant. As the figure showed, for rice, we used to find many kind of phytoalexins, but we focus on alexin A, B, C, and also saccharolantine. This is the phytoalexin response about rice blast effect. And after the treatment 5 days, we collect the leaf with this rice blast symptom, and then we extract to check by TLC method. And as this figure showed, the presence of possible phytoalexins of saccharolantine at IF volume 0.08 and alexin B at IF value 0.28 is we found in treatment by nanoparticle solution treatment in TLC plate. And we also need to check the HPLC, the exactly volume. But this is the preliminary checking. We can prove that our nanoparticles induce phytoalexin produced by rice. So that's why after that many kinds of nanoparticles were proved to against the disease, the plant disease, and as formulated as elicitor to induce the plant immunity to against the pathogen effects. And also, we have already done many kinds of particles formulated in elicitor as Dr. Kasim Soiton do it already in the marketing cell. But we still doing different kind of particles against the pathogens. And the nano elicitor for disease immunity is the elicitor consists of metabolites like ketoglycogen C, ketomino, and luteolins, which are antibiotic substances produced by ketamine species, plus nanocatosan and potent microorganism of bacteria Rhodopseudomonas species. And the properties of this elicitor is induce disease immunity in plants, stimulate the phytoalexin production in plants to against plant pathogens. And at the same time also stimulate the new root growth and revitalize the soil and make available plant nutrient for plants and increase plant ability to absorb nutrients and increase the yield. As we done in the rice, we checking the root length, root fresh weight. We get more high weight than the even higher than non-inoculated control. And application as liquid just spray. So this figure we also showed the nanoparticles of pure compound of luteolin and luteolinum, the chemical structures, and also scan the nanoparticle scanning electron microscopy. And this figure is the most smaller size, smaller as 170 nanometer in average. Okay, that's all my presentation. Thank you very much for your attention. Thank you.
lên lên nó đổ rồi còn cái cái cam cho nó chạy nhá. Để vào lúc giờ đấy. Đấy. Đừng để xa quá thôi, đừng để xa quá. Đấy. Nó bảo luôn cái nắp ở trong sọt đấy. Lấy bơm lên. Rồi, tôi có tải lại, rồi. Kéo. Nữa đi, em để cho con tải chạy ra nữa đi. Đấy, lau đi. Hãy subscribe cho kênh Ghiền Mì Gõ để không bỏ lỡ những video hấp dẫn. chưa đông nhỉ ạ? Cái này sợ cũng chưa đông.
Không có thử nè.
Không, cái này về mặt nó chỉ xe thôi, bắn á.
Không vẫn được mà. Xe vẫn được ấy.
Đừng mắc mạnh quá. Cái này
Meter,
bắn, cái này
meter,
cho bắn này.
meter, in a meter.
Ồ, đấy. Ok.
In a meter.
Từ để tí nữa quay lại xong, em để riêng đấy.
I think like it should be two half.
Dạ,
One, two.
dạ, dạ, dạ.
Two half. One half not enough.
Yeah.
Half only, eh? One, two.
Yes, you hit that. colleagues and organizer of the conference. Thank you for the invitation. And let me present the results of research on the use of grow regulation. Modern agriculture is impossible without the use of plant growth regulation in practice. Studies have shown that many plant regulators are physiologically active substances, increase in yield, product quality, improving fruit set, and reducing the overpairing. In addition, these substances have an impact on the drought and frost resistance of plants, as well as contribute to the increase of non-specific immunity. Recently, a large number of new generation fertilizers have been appeared. The study of the effect of plant is currently interested. In this regard, the study of responsiveness mechanism of tangerine plants to the use of growth regulation is very relevant and is provide a theoretical and practical significance. The objective of research finding that were provided obstacle phytoregulator for plant growth, Sileplant, silicon-containing universal fertilizer, and microbial nanoelicitor, natural product regulator for plant immunity induction and growth, was affecting for the content of synthetic pigments, slow chlorophyll fluorescence induction, and content of phytoalexin. Varieties are draft tangerine Meagava vase, the Ponceros triforata also rootstock. Tangerine is grown at plantation of subtropical scientist center, it's Sochi, Russia. The experimental scheme included 3 options as exogenous growth regulation: obstacle, nanoelicitor, and Sileplant. The control was done by spraying with water. Obstactin is a phytoregulator for plant growth which related to eoxin. Active substances is the potassium salt of naphthyl acetic acid. Sileplant is a silicon-containing fertilizer. Nanoelicitor. Nanoelicitor is constructed from active metabolites of hitomium cupreum and nanohitozan using electron spinning methods by Dr. Kasem Soitong, which applied for increase to diseases immunity. Foliar spraying during the growing season was carried out for 3 data at over phase. First data late May, beginning June, second late in June, and third spraying before 1 month at harvest in September. A result showed that the treatment of plants with growth regulation, obstactin, Sileplant, and microbiological nanoelicitor didn't affect the content of green pigments in the leaf. It revelates that the treated plants with Sileplant gave the highest number of carotenoids increased in the leaves and followed by nanoelicitor. It acts as the activation of protective reaction of the citrus plants. Also, there are reported on other factors involved. The soil and climate conditions of the Krasnodar region are unique, favorably distinguishing this region from other territory of Russia. However, in this territory there is a systematic negative impact of various climatic stress factors of fruit plants. For tangerine plants, limiting abiotic stressors are frosts in the absence of shoal cover in the winter, droughts, and elevated air temperature in the summer. At the highest temperature, the tangerine reduces photosynthesis. There is an inhibition on vital process and active ovary fall. Such finding on slow chlorophyll fluorescence induction expressed higher viability index in Sileplant than nanoelicitor and obstactin as comparison with the non-treated control. Photosynthetic activity index showed that nanoelicitor and plant gave significantly highest activity than obstructin. Photosynthetic activity coefficient showed significantly higher in nanoelicitor, plant, and obstructin than the control. So, studies revealed that the treatment of plants with this growth regulation didn't affect the content of green pigment in the leaves. It can be explained that the participation of the pigment apparatus in the stability and adaptation of plant indirectly related to the protective effect of carotenoid. For example, the suppression of express energy of triplet states of chlorophyll and singlet oxygen. Treating plant with nanoelicitor and plant increased in the number of carotenoids in the leaves, which contribute to the activation of protective reaction. With this, the activity of photosynthetic process determines the productivity, resistance to stress factors, adaptability, and viability of plants. The constant companion of photosynthesis is chlorophyll fluorescence, and its level depends on the intensity of photosynthetic process. Weak fluorescence is a characteristic of a viable photosystem, and strong fluorescence is characteristic of a weakened or inhibited photosystem. We will define the main parameters of chlorophyll fluorescence. These parameters allow us to access the functional state of the plant's photosynthetic apparatus. Signs change in the fluorescence directly or indirectly relate to all stages of the light phase of the photosynthesis process. It's shown that treatment with growth regulation led to an improvement in the state of photosynthetic apparatus and in general the functional states of wild plants. Treatment with regulator led to a smaller weakening of photosynthetic function, which was expressed in the higher value of the viability index, especially in the variants with the introduction of nanoelicitor and plant. Nanoelicitor showed significantly highest growth of plant and followed by plant obstructin. Plant treatment showed significantly highest mass of fruits and followed by nanoelicitor and obstructin. The highest diameters of fruits was shown in plant treatment and followed by obstructin and nanoelicitor as a result. The calculation of growth parameters and the number of fruits left on the tree during treatment with growth regulation was made. The active growth of shoots on the variant with all growth regulation was determined. A result also showed that the treated citrus leaves with nanoelicitor derived Hithomonium cupreum are found scoparon, a phytoalexin associated with resistance of citrus to some diseases. For example, associated with resistance of citrus rot or of Phytophthora citrophthora. It is noted that the processing plant, nanoelicitor and plant increase the number of carotenoids, terribly in changing the defense reaction. Dear colleagues, it's all, and thank you for attention.
Hello everybody, very good morning all of you. Myself Dr. V. Gopi Krishnan, Scientist C at Center for Drug Discovery and Development, Satyabama Institute of Science and Technology, Chennai, Tamil Nadu. So it is in India. Today I am going to talk about exploration of earthworm caste associated actinobacteria for plant growth promoting properties. Before I am going to start my talk, I would like to express my sincere thanks to Dr. Kasim sir and his team and the organizing committee for inviting me as a speaker and chair the session. It is a golden opportunity for me, and thanks for the opportunity. So the actinobacteria. So actinobacteria or the gram-positive filamentous bacteria. So it is rich with high G plus C content. So it is a beneficial prokaryote, and it is the treasure house for the secondary metabolites in terms of antibiotics, and it is mainly present in deep sea, tall mountains and soil, and especially extreme ecosystems. And the main metabolism of this actinobacteria is it can utilize any complex substrates, and it can synthesize some complex structures having the bioactivities like antibacterial, antiviral, anti-TB, and anti-cancer. The another important thing, commercially available antibiotics in the market. So most of them, around 80% of the antibiotics mainly produced by actinobacteria. So this is the unique nature of actinobacteria. In another way, it is also called as actinobacteria are antibiotic generators. Another important thing, it is one of the largest phylum in bacterial domain. In earlier period, in 1940s, only 5 genus at the time of streptomycin discovery. But in nowadays, it is having 6 classes, 46 orders, 79 families, 425 genera. So this is the largest phylum in bacterial domain. And regarding the natural product discovery, this is a recent report where you can see the antibiotics and all bioactive compounds produced by microbial sources, especially actinobacteria, eubacteria, fungi, and when compared with other fungi and bacteria, actinobacteria produced 20,000 bioactive compounds till now. So maximum of antibiotics commercially available. It may act as antimicrobial and anti-cancer. And not only antibiotics, actinobacteria, it is a paramount source of biologically important metabolites. It may produce some nanoparticles and solubilize the phosphorus, and it can produce some pigments. So natural pigments are need of the hour. So scientists are seeking for the natural pigments because in food industries, some of the synthetic pigments are very toxic. So replacement of that type of applications. Why don't we explore actinobacterial pigments for the food applications, food colorant? In addition to this, it is also, as you know, it is very much helpful for atmospheric nitrogen fixation, and it is act as a biocontrol agent and supported for plant growth promotion, and it can produce probiotics, biofiltrator, and novel enzymes. And it is also supported for soil amendment agent. So, doing the research on to isolate the novel compound, why don't we explore the extreme ecosystems instead of searching the same ecosystem for the novel compounds? So we can explore the extreme ecosystem, especially deep sea, ocean, and desert, and even very recently the areas like Arctic and Antarctic. The places were filled with ice. In these extreme conditions, the microbes are fighting for nutrients because these are the areas where having the very less nutrient content. So microbes are fighting for the nutrient. In that fighting condition, it can produce some novel compounds. That compounds may act as antibiotics, and it may produce some biofertilizer and probiotics also. So these are the unique nature of extremophiles. So we are doing our work on bioprospecting of actinobacteria from less explored ecosystems, and our team, myself and Dr. Radhakrishnan and Dr. Jareen Joseph, and our scholars, Mr. Manikundan and Ms. Abhirami. So we are all as a team, we are working on rare actinobacteria for the novel compounds So this slide you can see the earthworms. You all know that earthworms are greater friend of our farmers, especially for the agriculture area. So it is playing the major role in agriculture field. Uh, and it is also supported for the soil porosity, water infiltration and decomposition of organic matter, and the humus formation and suppression of soil-borne diseases and pests, nutrient recycling, cycling, plant growth, and these are the major role of earthworms, and it is having a different types. Red earthworms, blue earthworms, and we have we can see the different type of earthworms in the agriculture soil, and this is the article where we can think about earthworms in the high value manner. So the thing is, earthworms could help to reduce the antibiotic resistant genes in soil. As you know that antimicrobial resistance is a very big problem in medical field. Scientists are still having the very big problem because of the antimicrobial resistance. Still now there is no proper solution for antimicrobial resistance. So, but in these articles, they showed the very big important role of earthworms, and it is a powerful nature of earthworms could help to reduce the antibiotic resistant genes in soil. So, how much powerful earthworms are? So, this is what we have taken our work on earthworm, especially in the earthworm cast. So, instead of disturbing earthworms, why don't we explore the earthworm cast? And recently we started our research on earthworm cast. As you know that it is rich with the nutrient, and it is a nest of earthworm. It is filled with microbiomes, especially bacteria, fungi, and very particularly actinobacteria. So it is having the greatest health benefits for the plant, and like plant germination, growth, and increase the production. And it is also rich with microbiomes and provides a healthy root formation, and also makes plant healthier and increase the plant growth and increase the yield. So these are all the health benefits. So earthworm cast are the major research area. We started our work on earthworm cast, and especially the bacteria called actinobacteria, and we have started our work on isolation of actinobacteria from earthworm cast, and we have collected the material from agricultural soil around Kanchipuram city, Tamil Nadu, it is in India, and using the medias like nutrient agar, starch-casing agar, and cellulose agar. So you can see the beautiful pigmented colonies, different morphologically different colonies of actinobacteria and bacteria. So after incubation, morphologically different bacterial colonies were observed in all the three plates, three media. So, and we have isolated around 56 morphologically different bacterial colonies. In that, 36 are belongs to Streptomyces, that is actinobacteria. So all the actinobacterial cultures were subcultured in the media called yeast extract, malt extract, agar. So this is the basic media for culturing the actinobacteria. So you can see the different pigmented colonies of bacteria and powdery colonies of actinobacteria, and also in the last plate you can see the pigmented actinobacteria, that is pigments produced by the actinobacteria. And after the isolation, we have done the antimicrobial screening of earthworm cast actinobacteria by the method called agar plug. In that, we have screened the 36 actinobacteria for antimicrobial activity using the human pathogens like Staphylococcus aureus, Enterococcus faecalis, Escherichia coli, Klebsiella pneumoniae, and Pseudomonas aeruginosa. So you can see the after incubation, zone of incubation are around the agar plug. So this is the in this activity in the antimicrobial activity. Out of 36 actinobacterial stain screen, in that 12 isolates of Streptomyces were found to be actively showing the antimicrobial activity against one or more test pathogen. So this is the very important thing about the actinobacteria from the less explored ecosystems. So not only done the antimicrobial activity and also done the same kind of activity against plant pathogens, and also screening of indolactic acid, IAA production, and solubilization of phosphate, and enzyme screenings like amylase and cellulase, and also we did the taxonomy of the potential stain, actinobacterial stain. So for this purpose, we have selected 6 morphologically different actinobacteria from our culture collection. This table showing the different morphology of all the 6 stains, and after the isolation, we have done the screening. In the screening, we have done the activity against plant pathogen. So in this strains, only one strain, that is EWCA21, showed the activity against both clinical and also plant pathogen. So we have chosen this strain for our further study, and it is considered as our potential strain because of its broad spectrum activity by both for clinical and plant pathogen. And using the plant pathogen Ralstonia solanacearum, we have again done the triplicate, and you can see the zone of incubation. So you can see the plate of potential stain. So after that, we have did the effect of medium consistency and incubation period of bioptic components using the fermentation, both solid state and liquid state fermentation. When compared with the liquid state fermentation, it is showing the promising activity, and also yield also increased in the solid state fermentation. So we have chosen this for our further study. So after that, we have did the indolactic acid production. So the stain potential stain showed the indolactic acid production by cube method and also by 96 well plate method, and it is also solubilized the phosphate, and it also showed the amylase enzyme production and cellulase enzyme production. These are all the methods, standard methods we have followed, and also we did the production and extraction of bioptic compounds from the potential stain EWCA using the solvent called ethyl acetate. So we optimized the solvent extraction, and that ethyl acetate showed the better extraction. So after that, we have done the optimization of potential actinobacteria and different sources of media. So in optimization, we are preparing the media with different sources like carbon, nitrogen, and different pH and different temperature. In that, in optimization, carbon and nitrogen sources were increase the activity and also increase the biofertilizer production. You can see the activity in the down photograph, last photograph. So it is a taxonomy of our EWCA21 strain based on the characterization and taxonomy. You can see the microscopic appearance and the petri plate morphology. The potential strain belongs to Streptomyces. So based upon the characterization taxonomy, the potential strain belongs to Streptomyces. And our conclusion. So the study is basically the preliminary study. So the study revealed the strain EWCA21 earthworm caste associated actinobacteria will be an imperative resource for biofertilizer production and also for bioprospecting that could produce novel bioactive compounds useful for the agriculture applications. So still we are work under progress. We are doing the field level assays and productions, and also the assays, and recently we published the papers with the collaboration of Dr. Kasim sir, and his support is very big benefit for us, and still he is supporting. In future, we are going to do our research in collaboration with Dr. Kasim sir. We are going to screen the actinobacteria with the help of our Kasim sir, and also we are trying for some biofertilizers. So in future, definitely we'll do our collaboration, and also we can produce some biofertilizers and in cheaper manner, and it will be very support for agriculture persons. So, I would like to express my sincere thanks to Dr. Kasim sir and his team for the great opportunity. So it is a great opportunity for me, and thanks for the opportunity, and thanks for the organizing committee and all the uh, uh, delegates and scientists and the professors all over the world. So I'm happy to uh, see you all, and thanks for the opportunity, and save earthworms. So thank you, Dr. Kasim sir. Thank you.
Pleasant day to everyone. We express our gratitude for this year's 9th International Conference on Integration of Science and Technology for Sustainable Development. I am Elderico P. Tabal, a faculty of Western Mindanao State University, Buanga City, Philippines. Our deepest appreciation to be able to present our study on carbon emission equivalent of selected agroforestry systems in Buanga City, Philippines. The rapid increase in human population has placed a great strain on the world's soil resources. The continuing population pressure required the use of more lands to meet food demand, which had resulted in massive deforestation. The practice of till plant and fertilize cropping pattern has caused constant threats to the upland environments. These constraints are expected to escalate as food demand is expected to increase in years to come. The solution to the problem of providing enough food now depends on the extent of productivity level of our available lands, including those lands which are too steep to till but are currently used for. This prevailing problem can be addressed through the establishment of agroforestry systems being promoted within the community-based forest management, or CBFM, areas located mostly in the uplands of Zamboanga City, Philippines. The CBFM is a national program and the Philippines' major strategy for the sustainable development of the country's forest resources. Its evolution as a policy and practice in forest management that integrate and unify all people-oriented programs where the development, promotion, and establishment of agroforestry systems are exemplified in order to address poverty and environmental protection in the uplands of Zamboanga City. Food sources derived from agroforestry systems such as fruits and annual crops requires enormous energy to produce. Before reaching our plates, our food is produced, stored, processed, packaged, transported, prepared, and served. The entire process is fossil fuel based intensive. At every stage, there is high energy usage with high potential carbon emission equivalence. Hence, this study, with an objective to calculate the carbon emission equivalent derived from the energy inputs of the different agroforestry systems in Zamboanga City, Philippines. This figure shows to you the study sites, showing the map of the city of Zamboanga, Philippines, where on this map showing to you the 16 identified CBFM sites with a total land area of 12,406.6 hectares, located mostly in the hilly and mountainous portion of Zamboanga City, Western Mindanao, Philippines. And on these 16 CBFM sites, this table shows to you the list of dominant agroforestry systems identified and their tree and crop components across the 16 CBFM sites. There were 9 agroforestry systems identified and with their tree and crop components also showing in this table. In terms of energy use determination, the banana, tree, and annual crop components, their energy usage were determined and calculated from pre-planting operation, crop establishment, crop care and maintenance, harvest and post-harvest operations. Purposive sampling was used in selecting the fitted characteristics and identifying the actual respondents across the 16 CBFM sites. Subsequently, the identified 100 CBFM beneficiaries were interviewed as respondents using a structured questionnaire to determine the total energy inputs per hectare of identified agroforestry system within the 16 CBFM sites. Energy accounting procedures and coefficients were adopted from various literatures measured in mcal per hectare per agroforestry system. Then it was converted into little diesel oil equivalent, where 1 LDOE is equal to 11.414 mcal. That's according to Pimentel in 1980. Now, the total energy inputs is calculated in 3 ways. One, to get it from direct energy, second, from indirect energy, and third, from embedded energy. Direct use of fuel or gasoline to run machineries for farm operations and transport of farm products, that's the direct energy. When we talk of seeds, fertilizers like NPK used to fertilize crops within the AFS, and the agrochemicals, pesticides applied in labor fall under indirect energy. And finally, for embedded energy, it is on the utilization of machines, farm equipment, and implements, motorized vehicles, and draft animal use during farm operation. While the energy input for the manpower that includes food, clothing, and miscellaneous living costs of the farming household were not accounted. Table 2 shows the energy coefficients of various farm inputs. In this table, showing to you the various inputs like seeds used and agrochemicals, ranging from herbicide to insecticide used during farm operations per agroforestry systems. And also shown here the energy equivalents in terms of MCAL and references. There are also equivalent energy for chemical fertilizers and fuel, their MCAL equivalent units. So all these coefficients were used to account for the energy inputs. Finally, after having the total energy input in LDOE per hectare, it is multiplied by 3.96 kilogram carbon dioxide emission equivalents per LDOE. That's according to Pimentel, to get the net carbon dioxide equivalent, or expressed in tons carbon dioxide equivalent per hectare per agroforestry system. Now, for results and discussions, Table 3 shows the energy input in LDOE and the carbon emission equivalent expressed in ton carbon dioxide equivalent per hectare of the different agroforestry systems across the 16 CBFM sites in Zamboanga City, Philippines. So all of the 9 agroforestry systems accounted all the energy inputs from direct energy input to indirect energy input and embedded energy input. That makes the total energy input in LDOE and its total carbon dioxide equivalent per hectare in terms of carbon dioxide emission equivalents. The carbon emission equivalents described in this study is the amount of carbon dioxide emitted from the computed total energy input in the form of liter diesel oil equivalent during farm operations or consumed on a particular agroforestry system. The sum total of direct energy input, indirect energy input, and embedded energy input is called the total energy input that were computed from the various cultural practices and management, such as the pre-land preparation, crop establishment, crop care and maintenance, harvest and post-harvest operations. As shown in Table 3, the net C emissions or carbon emissions range from 2 to 4 ton carbon dioxide equivalent per hectare across the 9 agroforestry systems. Of this range, 94 to 98% of the carbon emission equivalent is attributed to indirect energy input, while the remaining 2 to 5.9% obtained from the direct energy input and embedded energy input combined. The results further showed that the multi-tree-based systems contributed to high energy usage, which explain why carbon emission equivalent is high, derived from indirect energy input, which is attributed largely to the use of inorganic fertilizers, agrochemicals, and labor. This is also called the energy hotspots. The energy hotspots refers to the high requiring energy activities or processes relative to the growth stages of a particular crop or tree species in a particular agroforestry system, accounted mostly in indirect energy inputs. This answers why over 94% of the emission equivalents attributed to indirect energy input across the 9 agroforestry systems. Hence, the indirect energy inputs contributed high in the total carbon emission equivalent. The high carbon emission equivalents was directly attributed to high usage of farm inputs such as inorganic fertilizers, particularly nitrogen fertilizer, herbicides, and labor, or call this the energy hotspots, which explain the increase in carbon footprints. Insecticide, for instance, was used primarily to control coconut scale insects infestation when this study was conducted, while herbicide was applied to avoid high cost on labor, particularly on weeding. This means that the tree-based systems were energy-intensive systems due to usage of nitrogen, phosphorus, potassium fertilizers, or call these chemical fertilizers or inorganic fertilizers, pesticides, and labor utilized from crop establishment to harvest and post-harvest operations, hence contributed high in the total carbon emission equivalents. The high plant density and the number of tree species present in the system contributed significantly in the overall energy inputs. This is due to the accrued energy usage that each individual tree crop components contributed, implicating the entire system to be net carbon emitter. Understanding the significant contributions of various energy-intensive systems delineated into direct energy inputs, indirect energy input, and embedded energy input will guide our local planners and legislators to initiate food production system with reduced energy and carbon footprints responsive to changing climates for the upland communities in the city of Zamboanga, Philippines. At this point, we'd like to express our deepest thanks to the Commission on Higher Education, the Philippine Council for Agriculture, Aquatic and Natural Resources Research and Development, or PICARD, and Yamag Bukid Healthy Products for the funding support for making this study a success. With this, I'd like to thank you for listening. Again, this is Elderico P. Tabal from Western Mindanao State University, Zamboanga City, Philippines. Thank you very much.
Our team works on the biological control of fall armyworm or Spodoptera frugiperda, which is a major insect pest of corn and other economically important crops. Specifically, our studies focus on the beneficial fungi called entomopathogenic fungi, which are effective biological control agents against various insect pests. What I will present today is an update on our attempt to formulate an indigenous EPF, Metarhizium malayi. I am Elisa Mendacalvo, and the rest of the team are John Rensara Macaraig, scientist Marcelo Navasero, scientist Mario Navasero, and Jose Maria Navasero from the National Crop Protection Center, College of Agriculture and Food Science, University of the Philippines, Los Baños. My presentation will tackle the background and objective of our study, what we did to achieve our objective, what were the results we obtained, and lastly, our way forward. This figure shows the Integrated Pest Management, or IPM. We know that possible damage to the pests can be achieved through IPM, wherein combination of crop protection solutions is being encouraged, with less dependence on synthetic pesticides for a more sustainable pest control. This slide shows the various IPM practices from pest monitoring, cultural, physical, biological, and chemical strategies. One of the eco-friendly measures to suppress pest population is through the use of biological control agents, such as entomopathogenic fungi. In corn, the fall armyworm or Spodoptera frugiperda is one of the most destructive pests. This polyphagous pest has a competitive advantage against other insect pests of corn. Since its first report in 2019, incidences were reported in various corn-producing regions in the Philippines, with infestation reports in sugarcane in 2020 and rice in 2021. This pest can damage the whole plant, from window panes and leaves, destroyed silks and tassels, to corn cob damages. Hence, management measures must be in place to prevent upshoot in its population. One of the IPM strategies against fall armyworm is through entomopathogenic fungi. But what are these? These are beneficial fungi that can cause significant mortalities and adversely affect the growth and survival of insect pests. EPF infect its host by adhesion to the cuticle, tissue invasion, enzymatic activity, and toxicosis. Metarhizium relayi is a cosmopolitan EPF reported effective against fall armyworm. Our first isolate of Metarhizium relayi was recovered during the outbreak of onion armyworm, or Spodoptera exigua, wherein we successfully isolated Metarhizium relayi from field-collected larvae. Likewise, during the fall armyworm outbreak, mycost larvae due to Metarhizium relayi were recovered in several cornfields. We discovered that the Metarhizium isolate from Spodoptera exigua was infective to Spodoptera exigua and cause-infected to Spodoptera frugiperda and putty armyworm, Metemna separata, in our laboratory setups. So far, we have tested the efficacy of crude suspension of this fungus against fall armyworm and other armyworm species. The production of viable and stable inoculum is critical in the utilization of this EPF. With the promising results in the laboratory, we geared towards cost-efficient mass production formulation of this fungus. Literatures suggest that oil-based formulations improve the persistence and insecticidal activity of EPF. Because oils serve as excellent stickers that promote contact between the formulated active ingredient and the lipophilic insect cuticle, as well as increasing rain fastness on the waxy leaf cuticle of treated host plants. Hence, this work was carried out to prepare an emulsifiable concentrate, or EC, of this fungus from Spodoptera exigua using various organic solvents and evaluate its bioefficacy against fall armyworm. We also determined the effect of these EC formulations on the mortality, growth, and development of the insect pest. To achieve our objective, the following activities were conducted in the laboratory from May to September 2022. The first step was mass production and formulation. Dry spores of the fungus were collected during full sporulation in artificial media. Subsequently, these dry spores were mixed with liquid carrier containing organic oil solvents and emulsifying agent. Spore concentration was determined using hemacytometer. Samples of these EC formulations were incubated in the refrigerator at 4 degrees Celsius, while other samples were kept in incubator with 25 degrees Celsius and 75% relative humidity. The test insect was also reared in the laboratory. To obtain significant number of third instar larvae for bioassays, adult moths were paired in mylar cases, supplemented with sugar syrup and healthy corn plants. Egg masses laid in the plants were collected in sterile Petri plates lined with tissue paper. Upon hatching, neonates were transferred to sterile pans and fed with fresh corn leaves daily. We then conducted bioefficacy experiments using third instar larvae. 3 tests were conducted. The first experiment tested the effect of oil solvents. The second tested the bioefficacy of EC formulations before storage. And the third experiment assessed the bioefficacy of these EC formulations after 3 months. The treatments were missprayed in surface sterilized corn leaves and an aliquot of oil solvents mixture similar to the volume of the EC formulation with Metarhizium relayi at 1 × 10⁷ cnidia per ml was prepared as spray suspension. Control setup were treated with liquid carriers and sterile distilled water. Treated leaves were introduced to third instar larvae that were cultured singly per Petri plate. Fresh, surface sterilized leaves were fed to test larvae daily. Experiments were laid out in completely randomized design, or CRD, and treatments were replicated thrice with 10 test larvae per replicate. In experiments, we gathered the following data: the larval mortality, pupation, adult emergence, and larval and pupal period. Mycosis of larvae was also noted. Analysis of variance was computed and means were differentiated in statistical analysis software using least significant difference, or LSD, with p-value less than 0.05. The following slides will highlight our findings on the effect of oil solvents, the bioefficacy testing of EC formulations before and after storage, as well as the effect on growth and development. The oil solvents used in the study was assessed for possible effect on fall armyworm. The various organic solvents did not cause significant mortalities to the test insect as seen in Figure 1. At 3 days after treatment, larval mortality ranged from 0 to 6.67%. Although there were mortalities recorded in other oil solvents, the values were similar with the control and did not increase significantly. This finding suggests that the oil solvents as liquid carrier had no insecticidal effect to the insect pest. Succeeding bioassays were conducted to compare the insecticidal activity of the EC formulations before they were stored and 3 months of storage in refrigerator and incubator. The mean larval mortalities were highest before storage, about 87%, while mean mortalities were lower after 3 months storage in refrigerator, 17%, and incubator, about 47%. These figures also suggest that all the EC formulations had similar effect, causing a 77 to 100% larval mortality before storage. However, mortalities generally declined after 3 months of storage, about 10 to 62%. Among the EC formulations stored in refrigerator, Metarhizium raleyi in mineral oil and sunflower oil caused significant mortalities. Those stored in incubator had a varying result, with Metarhizium raleyi in sunflower oil, mineral oil, peanut oil, and palm oil, inciting significant mortalities as compared to the unformulated fungus. These oil solvents also had the lowest reduction in virulence when larval mortalities were compared before and 3 months after storage. Those larvae that succumbed to fungal infection had stiff larval body. Mycosis of the dead larvae were also evident, signifying fungal infection due to the fungus. What fungal growth was observed and later turned to olive green as the fungus sporulated in the larval cadavers. Considering the little time, a slight difference was observed before storage of EC formulations. Little time in 3 months did not vary, which ranged from 3.75 to 9.5 days in refrigerator and 5.17 to 7.49 days in incubator. Based on the literatures, many factors affect the stability of the formulation, such as the genotype of the microorganism used as active ingredient, the formulation, temperature, relative humidity, and packaging. The loss of virulence of Conidia in the refrigerator is due to the delayed germination process, since the fungus undergoes the stationary growth phase and the growth rate is decreased. Previous research demonstrated that unformulated Conidia and formulations of Beauveria bassiana stored in refrigerator for 30 months had slow germination and low virulence. This is in contrast to other findings that Conidia viability of Metarhizium anisable var. Acridum did not vary between period of storage, temperatures, and formulations in the first 15 weeks. However, Conidia viability was significantly reduced over time, with remarkable decline between 35 and 40 weeks of storage, particularly at 27 degrees Celsius. This paper also presented that the various oils had varying performance as liquid carriers in the formulation and performed better than unformulated Metarhizium raleyi. The miscibility of oil in water suspension may also contribute in the bioefficacy of the EC formulation, since all of the EC formulations prepared have homogeneous mixture when mixed with water for treatment spray. Likewise, peanut oil, sunflower oil, and mineral oil, which do not solidify, appeared promising after 3 months of storage. Previous studies also noted that the type of oil used may affect the behavior of Metabasium relay and target insect. This fungus had good miscibility in sunflower oil between 80 and Tridonax 100, resulting in a uniform dispersion of conidia in water in its spray fluid. Likewise, another study confirmed that the oil used affected the insecticidal activity of Metabasium relay against fall armyworm. The effect of EC formulations was also determined in the growth and development of the insect pest, as shown in this table. The pupation, about 0 to 23%, was generally lower before the EC formulations were stored. High pupation was recorded at 70 to 90% due to lower mortalities in EC formulations stored in refrigerator. On the other hand, those stored in incubator, which incited significant larval mortalities, had lower pupation. A slight difference on the adult emergence was observed among treatments, regardless of storage time and condition. The larval period did not vary among the oil solvents and storage conditions. Likewise, pupal period was similar among treatments before storage and 3 months in incubator, while storage in refrigerator had slight effect in pupal period. These findings were consistent with our previous observations. This research work explored formulating a Philippine Metabasium relay isolate from Spodoptera exigua as EC formulation using various organic oil solvents as liquid carrier with emulsifying agent. It was evident that storage conditions affected their bioefficacy. It should be noted that this study employed varying methods in isolating the formulation, suggesting the differences in our findings with the previous results. Nevertheless, the efficacy and stability of these initial EC formulations under storage conditions are continuously being evaluated in terms of microbiological, physical, chemical, and biological properties. Optimization of the product is really necessary, such as adjusting the composition and process and understanding the behavior of the microorganism to provide an optimal development conditions and insecticidal activity. The authors greatly appreciate the effort and assistance of our research team. We also acknowledge the funding support from the Department of Agriculture, Bureau of Agricultural Research of the Philippines, as well as the support from our host institution, NCPC, CAPS, UPLP. Thank you very much for your attention.
A pleasant day, everyone. My name is Randolph Candano, and I will be presenting the results of our study entitled Preliminary Field Evaluation and Comparative Predatory Efficiency of Euborrelia species against Spodoptera frugiperda under laboratory conditions with notes on species characterization. I am a university researcher from the National Crop Protection Center, College of Agriculture and Food Science, UP Los Baños. This research study was conducted with the help and equally important contributions of my colleagues, namely Wilson Depanis, Mario Nabasero, Melissa Montecalvo, Joseph Manipol, and Marcela Nabasero. Fall armyworm, Spodoptera frugiperda, a relatively new pest in the Philippines, was detected in 2019 in the northern part of the Philippines. This caused heightened apprehension in the crop production, especially in corn. Moreover, this stirred various stakeholders in the country to acknowledge FAO as another serious threat in the brink. The resistance manifested by FAO against many pesticides available in the market adds up to pressing issues on crop production. Yu, in 1991, reported that a strain of FAO showed resistance at varying degree to pyrethroids that includes permethrin, cypermethrin, cyhalothrin, phenvalerate, tralometrin, bifenthrin, tetramethrin, and flubalinate, two organophosphorus insecticides that includes chlorpyrifos, methylparathion, diazinon, sulfropos, dichlorbos, and malathion, and to carbamates that includes metomyl, carbaryl, and thiodicarb. Zhang et al. In 2021 also reported resistance of FAO in varying degree against chlorpyrifos, spinosad, lambda-cyhalothrin, malathion, penvalerate, deltamethrin, emamectin benzoate, and chlorantraniliprole. The resistance manifested by FAO against many pesticides in turn places biological control agents in bigger stage geared toward to help manage its population. Among the commonly utilized biological control agents, or at least have been the subject of biological control studies against economically important pests, are the predatory earwigs, Euborrelia annulipes and Euborrelia annulata, but their capability as predator against FAO have yet been documented. The utilization of these Euborrelia as one of the options in managing pest population may not be hounded by great hindrances, as these can be simply mass-reared at low cost, which can be used easily and simultaneously. However, the area of concern is the accurate identification of the two predatory earwigs that are morphologically similar under the naked eye. The great resemblance of the two species often led to oblivious misidentification and confusion in terms of proper identification. Such dilemma was later resolved through re-examination of stock population of Euborrelia in the university. The objectives of this study was to provide aid in differentiating the two species for proper identification, to gauge the potential of the two earwigs in managing FAO in the field in comparison with commonly utilized entomopathogenic fungi Metarhizium anisoplay and Beauveria bassiana, to gather novel information on the predatory efficiency of the two earwigs under laboratory conditions against FAO in terms of number of preys consumed or slayed, prey size with third instar as smaller prey and the fifth instar as larger prey, and across sex to determine which is more aggressive in attacking and consuming prey. For the methodology, we have conducted the following activities: the differentiation of the two Euborrelia and sex identification, preliminary field evaluation and efficacy of the two Euborrelia against FAO, and predatory efficiency of two Euborrelia under laboratory conditions against FAO. Between these two predatory earwigs, Euborrelia annulipes is bigger than Euborrelia annulata, and the general difference between male and female of both species differed in the appearance of their cerci. Since Euborrelia annulipes is a bigger species, its genitalia is bigger than Euborrelia annulata. Precise method of distinguishing Euborrelia annulipes and Euborrelia annulata from each other using their unique morphological features based on the two works of Siribistaba in 1976 and 1979. The main basis of comparison is the presence and absence of elytra and the shape of the pronotum. Elytra and wings are totally absent in Euborrelia annulipes, while elytra is abbreviated in Euborrelia annulata, and wings are also absent. In terms of hind margin of pronotum, it is rounded in Euborrelia annulipes and subtruncate in Euborrelia annulata. This is the appearance of pronotum and mesonotum in Euborrelia annulipes and Euborrelia annulata. During the preliminary field evaluation on efficacy of the two Euborrelia against FAO, small plot experiment under field condition was conducted at Central Experiment Station, Pili Drive, UP Los Baños from April 11 to July 31, 2021. First half of the period is dry while the latter half is wet. The 5 treatments were E. annulipes, E. annulata, M. anisoplay, B. bassiana, and control applied weekly in a 5 by 4.5 meter plot with 6 rows of open pollinated corn variety. The 4 middle rows were designated as sampling points with a total of 20 plants per plot as sampling unit. A total of 22 earwigs were released per designated plots and rows, put singly in the world together with a little amount of wet coconut pit. Suspensions of M. anisoplay and B. bassiana with 10 to the 9 spore count per ml with talc as spore carrier were sprayed in all plants in the designated plots following the conventional manner, and amount and excess suspension were properly disposed. Data gathered were as follows: rate of leaf damage, count of damaged tassel and ear, and the yield data. Percent leaf damage was determined using a simplified Davis rating scale with 0 to 9 rating with the following descriptions. For predatory efficiency of Tuyo borrelia under laboratory conditions against FAO, third and fifth instar FAO were used as sprays in separate setup on May 5 and June 21, 2021 respectively. Clear disposable Petri plates were used as arena. With cannibalism, single prey was placed in each Petri plate prior to release of predator, and right after that, another prey was placed. Corn leaf discs with diameter of 35.25 mm were provided to preys to ensure that their behavior will be as natural as they were while being exposed to predators and to avoid death due to starvation. Leaf discs were placed on top of moist flattened cotton to prolong freshness. Soft paintbrush was used in transferring the preys to avoid injury. All predators used were starved for 24 hours and released in arena singly. For third instar prey, a total of 40 adult individuals for each predator, 5 females and 5 males were used in the experiment in 4 replicates. For fifth instar prey, a total of 50 adult individuals for each predator, 5 females and 5 males were used in the experiment in 5 replicates. Observation was 9 hours done from 9 to 1800. Each killed preys were recorded with corresponding time to determine its distribution within the observation period. Based on the results of a preliminary field evaluation on efficacy of the Tuyo borrelia against FAO, there was no significant difference in leaf damage, same with the number of damaged ears for all treatments. However, significant difference was found in the number of damaged tassels, wherein treatments E. annulata, M. anisoplay, and B. bassiana recorded lesser damage as compared to E. annulipes and untreated control. In terms of estimated yield expressed in tons per hectare, the 4 treatments, E. annulata, E. annulipes, M. anisoplay, and B. bassiana were significantly higher than the control. Among the treatments, plat release with E. annulipes and E. annulata produced the highest estimated yield, followed by plats sprayed with B. bassiana and M. anisoplay. The estimated grain yield for each treatment, E. annulipes, E. annulata, B. bassiana, and M. anisoplay were 26.13%, 25.31%, 13.37%, and 7.41% higher than untreated control respectively, which also correspond to higher income. With third instar FAO as prey, a total of 104 with a mean of 2.60 preys were killed by 40 adult E. annulipes, while a total of 77 and with a mean of 2.02 preys were killed by 38 adult E. annulata, as 1 female and 1 male died in the course of the experiment without killing any prey. The results showed that E. annulipes was more aggressive in killing preys, and the difference was significant. Of these, 73 and 51 preys were killed by females, while 31 and 26 preys for the males of E. annulipes and E. annulata, respectively. In average, the female killed 3.65 and 2.68, while male killed 1.55 and 1.37 for E. annulipes and E. annulata, respectively. Highest number of third instar preys killed by a single predator was 7 by both female E. annulipes and E. annulata. Between the two species, there was significant difference in the number of preys killed by female E. annulipes compared to E. annulata, but not in males of the two species. Most of the third instar being small preys were killed even in a single attack by both predators, and most of these were fully consumed by the predator before attacking another prey. A similar results were gathered using PIF instar prey, but the number of preys killed by both predators dropped immensely, a total of 60, with an average of 1.20 preys were killed by 50 adult E. annulipes, and 26 with an average of 0.52 preys were killed by 50 adult E. annulata. Results showed that the larger E. annulipes killed more larger preys than E. annulata, and the difference was significant. Of these, 52 and 19 with a mean of 2.08 and 0.76 preys were killed by females, while a total of 8 and 7 with a mean of 0.40 and 0.35 preys were killed by the males of E. annulipes and E. annulata respectively. Between the two species, there was significant difference in the number of preys killed by female E. annulipes compared to E. annulata, but not in males of the two species having identical values. Highest number of fifth instar preys killed by a single predator were 5 by E. annulipes and 2 by E. annulata, and both were females. Similar also in using third instar, results showed that the larger E. annulipes killed more fifth instar preys than the smaller E. annulata, and females for both predators killed more preys than males. All of the killed fifth instar preys by both predators were not fully consumed, leaving huge chunks of leftover cadavers. The huge drop in number of preys killed by both species can be attributed to the size of the prey and its instinct to thwart the attack initiated by the predator. Therefore, single strikes were not able to cause immediate death of the prey, but through series of attacks by the predator, the prey weakens and became defenseless, providing opportunity for the predator to initiate its killing strike. Hours were substantial windows for E. annulata, which registered highest number of third instar preys killed, but for E. annulipes, it was the first 3 hours that registered consecutive highest number of third instar preys killed. This could be attributed to 24-hour starvation that ensured drive for prey hunting by the predators. The first 2 hours for both predators was also substantial, as it reflected the ability and the tactic in killing their fifth instar preys. Highest number of fifth instar preys killed by both predators was recorded in the second hour and only 1 each in the first hour. This inferred that the aside from starvation that pushed the predator to hunt for prey, the size of the prey also played crucial role in the incidents. The larger fifth instar preys are not only stronger than third instar preys, it also retaliated against the attack of the predators. Hence, the predator needed multiple attacks to kill the prey. After the third hour, the general trend for prey consumption of both predators showed a drop and then an increased trend again after an hour. The habit perhaps can be explained by animal instinct that once the predator is full, it tends to be less aggressive in attacking prey and regain aggressiveness if starved. The experiment also showed that even Euborrelia are nocturnal, multiple hunting in daytime can also be extensive for these predators. This is a plus factor for predators being tapped as biological control agents in crop production. Euborrelia are known to be voracious predators, and its potential as biological control agents were cited by various researches and literatures. In the Philippines, potential of Euborrelia annulata as biological control agents against Asian corn borer was cited by Javier and Rey Jesus, and against rice blackbag as cited by Navarrete and Barrion. However, citation for the potential of Euborrelia annulipes as biological control agents in the Philippines is lacking. The lack of citation for Euborrelia annulipes is possibly due to the following reasons. First, government institutions knew all along that they only have Euborrelia annulata that is being promoted and distributed to end users and is being used in research purposes in the country. Second, the stock population of these institutions assumed to be Euborrelia annulata might be unknowingly mixed culture of Euborrelia annulata and Euborrelia annulipes. Hence, even when Euborrelia annulipes have been used for various purposes, it was acknowledged as Euborrelia annulata. With this, intensive information dissemination is warranted in the country to purify stock population of Euborrelia, and if needed, to optimize mass rearing and utilization of these Euborrelia species. The simultaneous use of the two predatory earwigs with the two entomopathogenic fungi is not strongly suggested for now. Further investigation must be conducted first on their simultaneous application, as the two entomopathogenic fungi may have significant adverse effect against the two predatory earwigs, thereby defeating the purpose or perhaps no significant effects at all. And lastly, apart from the efficacy of the four biological control agents, especially the two predatory earwigs, their low production costs can be considered in a decision to include these as components of integrated pest management against fall armyworm. That's all. Thank you very much. For more information, you can visit the following references.